The single-celled protozoan parasite, Entamoeba histolytica, is the primary infectious agent responsible for severe amoebic dysentery in humans. The taxonomy of the organism describes exactly what it does to a host. The term histolytica literally translates to tissue destroyer. This specific species operates as a monogenetic parasite. It requires only a single human host to complete its entire life cycle, without relying on secondary animal vectors. This creates a biological problem for the organism. To successfully spread, a fragile, microscopic cell adapted to dissolving human tissue must somehow survive exposure to the harsh, nutrient-deprived environment outside the body. The solution is a strict dual-state architecture. The parasite alternates continuously between a dormant, armored infective phase and an active, vulnerable invasive phase. This diagram outlines the biological life cycle of Entamoeba histolytica, centered around the human digestive tract. Following the red and blue arrows reveals a perpetual loop. The pathogen exits the host, survives in a dormant state, and is ingested to begin again. Neutralizing this pathogen requires tracking these precise cellular transformations. By looking at how its morphology changes at each stage, we can understand how it dictates its destructive capability. Human infection begins through direct contamination. The host ingests food or drinking water that has been compromised by the parasite. Common mechanical vectors in the environment, specifically houseflies, frequently act as agents transferring the pathogen onto these food sources. The specific unit swallowed by the host is the mature quadrinucleate cyst, illustrated here with its 4 distinct internal nuclei. The defining feature of this phase is its thick, transparent outer wall, providing extreme resistance to adverse conditions like stomach acid. This biological armor allows the cyst to pass safely through the upper digestive tract until it arrives in the large intestine, a nutrient-rich and highly favorable environment. Upon reaching the colon, the organism undergoes excistation. The dormant mass actively forces itself out of the protective shell through a minute pore. Immediately after emergence, this metacystic form undergoes cellular division, splitting into 4 active feeding uninucleate amoebulae. The dormant outer shell has served its purpose. It bypassed the host's external and gastric defenses, unleashing the active feeding generation directly into the intestinal cavity. This adult, actively feeding form of the pathogen is officially known as the trophozoite, or the magniform. In this diagram of the trophozoite's morphology, the cellular layout is divided into 2 distinct layers, a clear ectoplasm and a dense endoplasm. Suspended within are a spherical nucleus and food vacuoles. For clinical diagnostics, the critical markers are these ingested red blood cells. Discovering red blood cells trapped inside the parasite provides definitive proof to a pathologist of the organism's histolytic, tissue-destroying nature. The parasite executes this destruction chemically. It secretes an enzyme called histolysin, which dissolves the human intestinal mucosa on contact. While the trophozoite feeds on the dissolved tissue, it multiplies rapidly along the wall of the large intestine using simple binary fission. This diagram illustrates the systemic threat. If multiplying trophozoites breach the local intestinal mucosa, they enter the host's bloodstream. Following the invasive pathway upward shows secondary infection sites. The pathogen can travel to the liver, lungs, or brain, producing lethal abscesses. The trophozoite's internal chemistry is entirely optimized for biological extraction and tissue destruction.
However, this active form is incredibly fragile and cannot survive exposure outside the host. To propagate to a new host, the pathogen must eventually abandon its active feeding state. Certain dividing trophozoites initiate a severe morphological shift. They become smaller in size, round in shape, and noticeably sluggish in their movement. This illustration traces the pre-cystic transition. The first change is the rapid disappearance of food vacuoles. The organism shifts its energy strategy, storing reserve glycogen granules directly in the cytoplasm. Simultaneously, dense rod-shaped protein bodies appear within the cell, identified here as chromatid bars. The parasite then executes its primary defensive maneuver. It secretes a transparent, highly resistant cyst wall that entirely encapsulates its body. This precise secretion event marks the boundary between the active internal pathogen and the environmentally stable survivor. By completing incestation,
the organism successfully trades its invasive chemical weapons for impenetrable biological armor. While the newly formed cyst is physically walled off, it is not completely dormant yet. Critical internal maturation must occur before it is capable of infecting a new host. Inside the sealed capsule, the single nucleus undergoes a modified form of mitosis. This first division results in the binucleate cyst. A second and final nuclear division follows, splitting those two nuclei to form four. This specific tetranucleate, or quadrinucleate stage, represents the final, fully infective form of the pathogen. During this locked phase, the organism sustains itself by slowly consuming the glycogen and protein reserves it stored during the pre-cystic transition. The mature cysts then move into the host's exit pathway, passing out of the body entirely through fecal matter. Deposited in the external environment, the cysts now lie in wait. Their thick walls make them highly resistant to adverse temperature swings and chemical changes. The biological life cycle is now complete. The organism has successfully reset its state, perfectly primed to trigger excistation the moment it is ingested by a new human target. Entamoeba histolytica relies entirely on alternating between two master forms, the invasive, tissue-destroying trophozoite and the dormant, environmentally resilient cyst. This creates a stark clinical reality. While the trophozoite is responsible for causing the devastating internal symptoms of amoebiasis, the disease can only spread to a new host through the resilient cyst. Because the parasite is monogenetic, it has no intermediate animal hosts to rely on in the wild. Without secondary hosts, the survival of the entire species is strictly bound to direct fecal-oral contamination between humans. This raises a practical question. How do you stop a microscopic threat that is practically immune to external environmental exposure? The most effective solution is not entirely pharmacological, but environmental. Basic prophylactic measures can physically break the transmission loop. At the societal level, improved sewage disposal acts as a physical barrier, strictly isolating the mature cysts from any subsequent human contact. Agricultural interventions, like strictly protecting local drinking water and vegetable crops from biological pollution, cut off the primary ingestion routes. And at the individual level, simple, careful hand washing with soap and water after using the toilet mechanically neutralizes the danger of transfer. Entamoeba histolytica is a master of internal morphological adaptation, but neutralizing its destructive single-host cycle ultimately depends on breaking the physical chain of contamination in the outside world.